astrocytes to try to solve the problem, whatever it is. But if they cannot solve the problem, then that's when things go haywire and neurons die off. And I encourage you to look into this mechanism of action further. So what causes inflammation writ large? Well, there are a lot of things. Here's a list. One is immune senescence. So immune cells, we know, decline in function with age. That enables microbes to run rampant. Tissue damage, such as chronic unhealed injuries, cause chronic low-grade inflammation. Sugar and metabolic syndrome. There are a lot of mechanisms behind this, but one simple one is the formation of advanced glycation end products. So proteins get glycosylated, and this results in those proteins binding the RAGE receptor for AGEs, which enhances inflammation. Gut barrier permeability is a big one. We'll talk about this, also known as leaky gut. Microbes and PAMPs enter the local gut stroma. That causes local inflammation, and then if it's bad enough, chronic systemic inflammation. Another one is DNA damage and the de-repression of transposons. These are endogenous retroviruses that actually make up a huge chunk of our the dark matter of our genome. Cellular senescence, this is a big one. You can go to one of my prior lectures to learn a little bit more about that. And this pro-inflammatory cytokine milieu called the SASP, senescence-associated secretory phenotype, that is responsible for sending out this pro-inflammatory signal to the immune system. It's a come eat me signal to the immune system. So senescent cells have undergone stress such as DNA damage, mitochondrial failure, et cetera, or telomere attrition, and they need to be cleared. This is a mechanism to prevent the occurrence of cancer. And it sends out this pro-inflammatory signal to the immune system to come eliminate the senescent cells. If the immune system is too aged or compromised in some way to get rid of the senescent cells, then you get this buildup of pro-inflammatory load. And so that's when the senolytic drugs come in. At Apollo, we have a portfolio company working on this, and there are a handful of others out there, developing drugs that will selectively ablate or eliminate these senescent cells. Another is autoimmunity. I don't pretend to understand this. I don't know if anyone does. This is sort of a catch-all term or a black box, but when the immune system reacts against self proteins. And then you have all of these other factors: stress, lack of exercise, lack of sleep, neurotoxins like heavy metals and pesticides and other agrochemicals, insufficient anti-inflammatory foods, and so forth. So there are many things that can cause inflammation. One that I want to talk about is the gut-brain axis. So it was said by Hippocrates, the father of modern medicine, that death begins in the gut. And traditional Chinese medicine and Ayurvedic text, writing some thousands of years prior to that, actually said, interestingly, we agree with Hippocrates on this one, I'm paraphrasing, and that they all argue that the strength and quality of digestion and the gastrointestinal system is critical to the aging process. Because, you know, the GI tract is actually outside of the body, technically. It's just a long tube, kind of like the internet. It's just a series of tubes, right? So, so that's a tissue that we really need to protect, and it's responsible for assimilation of nutrients. And when that process breaks down, we become deficient. Even if we're eating enough of some of these nutrients, if your gut is not absorbing them, that's a problem. And actually, in my own work in the lab, previously did a Fulbright fellowship in a lab working on telomere biology in zebrafish. And we found that the first tissue to decline when you knock out telomerase in the zebrafish is actually the gut. And that makes sense because the gut is the most proliferative tissue, or one of the most proliferative. Hematopoietic stem cells are also highly proliferative. And interestingly, when you knock out telomerase, that's really bad for the HSCs in the bone marrow too, as a side note. Another great thinker who called attention to the gut-brain axis, or the microbiome before it was cool, fashionable, was Elie Metchnikoff. He won a Nobel Prize for his discovery of macrophages and was co-awarded with Paul Ehrlich. He was one of the first biogerontologists as well. So he studied a population of long-lived Ukrainians. They attributed their longevity to fermented foods and yogurt. And so he was an avid yogurt eater for the rest of his days. And he did live a pretty long time, I believe. So he studied the gut microbiome before it was cool, and he knew a thing or two about the immune system. So maybe we should follow up on what he said. So what are the physiological mechanisms underlying the link between gut permeability and brain problems. Well, here's one. You have a bad diet and lifestyle. So you have high sugar, low fiber, lack of exercise. Maybe you're eating a lot of molecules that induce gut barrier permeability, such as gluten found in wheat. You can believe the hype. Gluten, even if you don't have celiac disease, in some proportion of the population, they have non-celiac gluten sensitivity. Gluten is a protein that punches a hole in your gut barrier in the one cell layer thick epithelium. It binds a protein called zonulin, pops open the tight junction, and then stuff gets across that membrane that shouldn't get there. So it's leaky gut syndrome. And you can look into the work of Alessio Fasano, who was at Harvard, who detailed the molecular basis for celiac disease. And he also says that gluten punches a hole in the membranes of pretty much everybody, even if you don't have celiac. Whether you get symptoms depends on your immune system and some of the MHC polymorphisms that you possess. So anyway, there's a lot of stuff that can cause gut barrier permeability. This can also cause dysbiosis, so the growth of bad microbes that shouldn't be there, like Candida albicans if you're eating a lot of sugar. This causes local inflammation, and that causes peripheral inflammation. If you have leaky gut, you have molecules such as food peptides from proteins not fully digested to their amino acid constituents that get across that membrane, and then they're recognized by dendritic and other immune cells. That acts almost like a vaccine. So when you have a leaky gut and you eat foods that are immunogenic at all, you become more responsive to them. And so you're basically vaccinating yourself against your own food. And that creates de novo food allergies and also autoimmunity if the peptide sequence of what you're eating resembles the peptide sequence of a self protein elsewhere in your body. So there's a molecule lipopolysaccharide. It's a PAMP that is detected by the intestinal epithelial cells. This activates our favorite protein, NFKB, which is a master regulator of inflammation. That results in secretion of pro-inflammatory cytokines like IL-1 beta. IL-1 beta, interestingly, increases both gut barrier permeability and also permeability of the brain because it circulates, right? It's a circulatory factor. So IL-1 beta and probably other cytokines are actually linking the permeability of your gut to the permeability of your blood-brain barrier. So the microbes and LPS and other PAMPs get into the brain, induce cellular senescence, neuroinflammation, and other bad stuff. If that persists, you get T cell infiltration, and that's really bad. And we'll get into that, so show some data on that. You get cytotoxic T cells coming in, and those T cells can actually poke other cells and tell them to apoptose. And so that is what we seek to avoid. If that happens, it results in neurodegeneration. So if the microglia can't do the job, then the big guns come in. These are the T cells, infiltrate the brain, and they start killing off neurons, as you can see in this confocal image here. So the dogma for a long time was that the blood-brain barrier keeps T cells and peripheral immune cells out of the brain. That they also believed that the brain was sterile, that there were no microbes present. Even in healthy people, we are finding microbes in the brain, especially if you are not healthy and you have barrier permeability, then we're seeing a lot more microbes in the brain. So one of the exceptions, one of the results here in 2019 showed that T cells, there's a paper I'll show you, showed that T cells repress neurogenesis found in the age hippocampus and the subventricular zone during memory loss. So this was a very cool paper from the lab of Anne Brunet, who is also a major force in the aging field. And she and others showed that T cells infiltrate the brain and specifically get into the neurogenic niches where new neurons are created. So the T cells get in the mouse brains, they go to the niches, and they secrete interferon gamma, which is sort of an antiviral response cytokine that is sufficient to inhibit neurogenesis both ex vivo in dishes and in vivo and compromise cognitive function. So as a brief anatomy lesson, at least in the rat brain, you have several areas of neurogenesis. In the olfactory bulb, because these animals have a really keen sense of smell, in the subventricular zone, and also in the dentate gyrus of the hippocampus. And between the olfactory bulb and the subventricular zone, from the subventricular zone, you have the rostral migratory stream. We don't have that in humans, or very little. So, and it's pictured here, the subventricular zone, right outside the ventricles. And one of the reasons there's neurogenesis right next to the ventricles is because the ventricles contain CSF, cerebrospinal fluid, that bathe these neurons in signaling factors, such as growth factors and neurotrophins that tell them to proliferate. So in that paper, Brene and others found that in young animals, there is not very much infiltration of these CD3 positive T cells in the, say, subventricular zone. But in old animals, there are more. Say you can see about 4 of these cells in this section here, and it's quantified there. So you're getting more T cell infiltration in aged brain. And also those T cells rife in the aged brain, they're actually releasing interferon gamma, and they're expressing, as you can see, BST2, this interferon gamma responsive gene. It's quite a substantial effect size. Also in post-mortem human tissue samples, you see more T cell infiltration. So in the young human, you see few CD3 and CD8 positive T cells, but in the old, you see more, and that is quantified here. Switching gears a bit, one of the most intriguing interventions in the aging space that's gotten a lot of press attention because it's kind of vampire-y and captures the public attention is heterochronic parabiosis. So you take the circulatory systems of an old and a young mouse and you link them such that they share blood. The old mouse gets younger, the young mouse gets older. The young mouse gets more old than the old mouse gets young, which indicates that there's actually more bad stuff in the old blood than good stuff in the young. A lot of this work was pioneered in the Bay Area at these institutions, and some of it is being taken forward by this Spanish blood products company, Grifols, now. And they recently announced some compelling data, not that recently, over a year ago, I guess, data finding in Alzheimer's disease that if you actually do plasmapheresis, you scrub out the blood and you just replace it with saline plus albumin, a blood component, it actually works quite effectively. One of the largest effect sizes we've seen, even in moderate dementia patients. So not most Alzheimer's trials are in mild patients. These are actually in moderate patients. And we're seeing a pretty decent spread in the data there. So it's a 60% reduction in progression, even in moderate AD. This comes out of the work of primarily Irina Conboy and Michael Conboy's labs, as well as Tom Rando and Tony Viscore's lab. Plasmapheresis, you scrub the blood. It's kind of like bloodletting, which has been done for millennia. There may have been something to it, but you replace the albumin. Sadly, this is not one single profitable drug, a pill you can pop, or an antibody you can inject. So pharma hasn't really paid a lot of attention to it because it's not scalable, it's not really patentable, but thankfully Grifols is. And by the way, I do own stock in Grifols, although not very much. Okay, so coming on to the amyloid hypothesis in Alzheimer's, I have a very substantial bee in my bonnet about the amyloid hypothesis, and hopefully you'll see it's for good reason.